we just formulated some questions, but maybe just first thinking about, what do you see as now the greatest challenge to discover new antivenoms? Maybe I'll just start. I'll deal with you.
Yeah, thank you very much for that question.
Just take the microphone.
I thought, firstly, as was highlighted during the presentations, the venom variability. It's a major issue. The fact that you are dealing with a cocktail mixture of several venom toxins and no one toxin in particular is the pathologic the motive, so to say. So the complexity is massive, and that means you cannot have multiple antibodies to each of these toxin epitopes. And one has to explore approaches where you introduce potency and also reduce redundancy and have better affinity and have a situation where weight for weight you have neutralization of the venom toxins in particular. These are some of the issues that I see as a major challenge.
Yeah. So maybe I'll just go.
Yeah, I agree with Abdul, but I think that the big challenge is that we will never have a global antivenom. So forget about it, because we showed many times about high variability between these venoms. But for sure, we want to find some antivenoms with cross-reactive and produced for specific regions, maybe one for Africa, one for Asia, one for Brazil, because of course these snakes have some similarities, and we should focus on these similarities to produce locally, not locally I mean in a country, but different antivenoms, but high neutralizing and with low cost.
Well, I don't think discovery of binders is going to be a problem at all. I think this is obviously a multi-decade project now, and I think we have the tools and technologies to make high affinity binders. Andreas talked about cost of goods and production. Certainly that's an issue. Cold chain was mentioned earlier, but also I think regulatory hurdles. If you think of the cost and effort of taking a single antibody in the developed world, the cost and effort of getting that through a clinical trial. The question then is, what happens when you've now got a cocktail of 5 or 10 of those antibodies, even assuming you can get over, well, I do believe we'll find a solution to make 5 or 10 antibodies in a mixture, but it's just then the effort to get that into clinical trial and approved. And I still can't quite figure out the how you move from one end of the spectrum, which is the cost of making antibodies in the developed world, to single targets, versus the fact you can take serum from a horse that's been injected with a variable amount of venom and still get to a position where you can inject that into a human. So hopefully we can find a pathway that sort of meets somewhere in the middle that actually makes it feasible and viable to pass clinical trial.
Yeah, I think from my perspective, there's lots of challenges. I'll add some new challenges to the mix rather than perhaps what the priorities are. But I think there's also a big gap in terms of if we achieve all of this and we have a whatever it is, whatever this product looks like for whatever reason, is also getting that therapeutic to the right people, the right time. We can't currently do that with antivenoms that have been around for over 100 years. So there are major challenges in terms of accessing drugs as well to the people who really need it. But on a more specific level, from an antibody point of view, when we're thinking about monoclonal antibodies and perhaps oligoclonal mixtures, what does that look like? But also, how are we going to clinically trial these in the first instances? So what does the incipient new antivenom look like? Is this, we just get one antibody that's good against certain snakes and we use it in combination with current treatment and see do we have a clinically beneficial effect over conventional antivenom? I think there are some challenges there as to how we actually demonstrate in a clinical trial the effectiveness of these antibodies too, given the heterogeneity you've all heard about with different venoms, different toxins, frequency of snake bite and the like of it. So I think there's a huge number of challenges on every level. And perhaps as John alludes to, maybe the discovery is not going to be the biggest challenge. Clearly it's the first one now.
And I agree. I also don't think discovery is a big issue. I think manufacturing is an issue, but that can be solved. I think really the big issue is clinical trials. I think we have an opportunity to actually do phase 1 clinical trials, which is not something that is currently done with antivenom because it has side effects. So I think we have both an opportunity, but also a huge challenge of exactly as you say, Nick, which products do we move into clinical development? How are they going to be regulated? I think it's this year that the WHO expects to have their target product profiles for non-serum derived products. They made target product profiles for existing type products last year. I hope that they will be reasonable. I fear they won't. I fear that they will be totally unreasonable and either move it completely over into expecting that this should be, you could say, regulated just as any cancer therapeutic. And if we end up in that end of the spectrum, then I think we have a challenge creating business cases, creating incentive for governments, for companies, for serum institutes to actually really invest in new technologies that I truly believe have the potential to deliver much better therapies. So I think it's regulatory. We have the largest issue, then followed by clinical, then maybe manufacturing, and then finding the binders. That's a piece of cake.
Yeah, so I've heard several times indeed that translation from the preclinic to the clinic is going to be critical. So what would you recommend? There's certain animal models. You've shown some pre-challenge models, therapeutic models with different snake venoms. So how are you going to approach them?
I think that the sort of the good thing when you test in any animal model is if you take a mammal, it typically resembles at least somewhat the human case. It's a little bit like antibiotics. If they work preclinically, they have a very good chance of working clinically. And I think that, I mean, I hope one of the simple things that we could help do is not request in journals that we have human data. And we accept that actually this is a little bit like antibiotics where people get super excited if you've shown something in a petri dish. But it's actually hard to move this science into the respected journals and get, and by doing that, it's not so much to get the credibility amongst other scientists, it's actually really to get interest from governments and investors that look at this. And that's a simple challenge that people here in this room could help solve. It's simply just stop being so complaining as reviewers. I'm not saying any of you particular, but damn, it's hard to get some of this stuff out. And then you see people publishing, I'm sorry to say, not very good stuff in other fields where they get super excited. One person has this disease in the world, bang, into Nature, okay. There's 150,000 people that are dying here and it's being neglected and it's being forgotten because it's not prestigious, like I think it is with many other neglected tropical diseases. So sorry for being blunt, but I actually think this is a problem we could easily solve that would help things.
So maybe what do you think about the models that we should use to?
Well, definitely we'll continue with the animal models at the beginning, but eventually there has to be use of these drugs in humans, even if it's small number of people. And I guess with the newer agents, you'll have more safety and less early adverse reactions and less mortalities. And so you may be able to introduce them into traditional phase 2 studies in a few group of people, 5, 6 people, establish that they are efficacious, and then subsequently bring them into a larger group. Definitely will not see much use of placebo control trials in drug development for snakebite, largely because you're having the environment or that is associated with case fatality rates that will be unacceptable to ethics committees and IRBs. And from the point of view of the initial introduction into humans, I also foresee other designs coming into play. The adaptive design, for instance, is already gaining ground, and authorities are considering it much as a better design than the continual reassessment method using 3 people and then adding another 3 that I mentioned. Another interrelated problem that I see is endpoints. Even if you try it in a group of 6, what endpoints, what outcome measures are you going to use? If you are working in a place where the snake fauna is really very variable, you have some causing cytotoxicity, some neurotoxicity, some causing coagulopathy, you will not have a defined endpoint to outcome measure that will be reproducible through the range of all the patients. So we have to think of biomarkers, and we have to also explore standard ways for measuring the same kind of outcomes in the future.
Okay, I also want to open it up to the audience. So any questions you have for the panel or a specific speaker, please.
Some of you, I think John mentioned Adam 17 as a metalloproteinase, well, human. Is there a possibility that obtaining the antibody against snake metalloproteinase would actually block human metalloproteinases, because they are quite identical? Some of the active sites are almost identical. In a sense, you would not see this from maybe a model.
I think that's possible. I think that would also be true of not just of the antibodies, but the small molecule inhibitors that have been produced in the past. But I think if you contrast the toxicity of a snake venom with the toxicity of a kind of off-target blockade transiently of an endogenous protease, I kind of feel the balance would tip in favour of getting through the acute phase of snake envenomation. I think on the scale of things, that's a far worse problem, and people would probably accept some level of toxicity that might not be acceptable in oncology.
Yeah, I was just going to add to that. I think the risk benefit's quite different, and there are quite a few matrix metalloproteinase inhibitors that went into phase 3 for other indications and were reasonably well tolerated, but there you're looking at chronic repeat dosing over many months. Whereas for snakebite, you clearly would have a shift, and obviously from an acute versus chronic treatment perspective, there's a bit more leeway there too.
Yeah, just a simple add-on. It could, of course, happen that when you have a monoclonal, that it just is so powerful that and if it by coincidence cross-reacts. But I think at least to me, comforting thought is that existing antivenoms do target snake venom metalloproteases, and to my knowledge, we don't see any issues with this. Of course, you could say the antibodies are dilute, so there might be one very potent that does, but it's not my largest concern.
So I'm really new to this snake antivenom field, but I was wondering how what is the human immune response against these venoms? So because I don't hear you about B-cell sequencing or a repertoire analysis or these kind of things. So what's the status there?
So Jake Glanville, I know, has been working with some guy that injects himself, or he allows himself to be bitten regularly by snakes. Sorry, is that what you were going to say? Yeah, you're going.
No. Yeah, of course, they can be immunogenic, but some toxins are very small and they are not so immunogenic. And we have this famous guy that injected many venoms, and he was a little bit resistant for some. But I think the main thing is that when you receive the antivenom, you will block, of course, the immune response against venoms. And but we are trying to do some research in the Yanomami community, as I told you before, because our indigenous, many of them, they survive without antivenom, and we think that they can get kind of resistant because some of them have been bitten 3 times, and for sure we can induce an immunological response and a memory response against. But actually we need to understand more this, and I think you were asking about possible vaccine. But before we discuss a little bit, that would be very expensive, and for vaccine a longer and a big population would be not so. I don't think we could spend money on that, but I can hear other.
Well, I know Australian group working in Sri Lanka have also looked at that closely. There is cytokine storm at the beginning. You could have IL-6 elevated, also complement perturbations in tryptase, and then following administration of antibody, you have some coming in with anaphylactins, C5a, C3a, among other things. But this sort of, in time, it's attenuated. In addition, for the tissue pathologists, as we've been working with Nick Casewell's group, we've seen other cytokines in addition to IL-6, things like damage-associated macrophage protein within tissues, within blister fluids, among other things, are also elaborated. And of course, metalloproteinases are also seen. Human metalloproteinases are also released within the blister fluids. Yeah.
Yeah, I think another question is the format of the antibody. I think we talked about polyclonals, fragments, whole antibodies. How do you see that? Maybe some smaller fragments could be better go into a tissue where you need it, or maybe for some applications you need larger antibodies that have a longer half-life. How do you see that? What would be, what are the issues we need to work on?
I think that it is a good idea to have at least one format that has a long half-life that can, as you saw Dr. Habib show earlier, that you had these people that got the Fab antibodies, they reduced their symptoms, then some hours went by, the Fabs were cleared, suddenly they had coagulopathy again because more toxins have likely exited the bite wound. So I think whether it's exactly IgG or nanobody with an FC region or something, I do think that's a good idea to have in most cases. But I also think there's a chance that it might be relevant to have nanobodies or small molecule that can more quickly diffuse into deep tissue and neutralize. Exactly what the ratio between those, it could be the same nanobody that exists both as naked and as FC fused, I don't know. But I think there's a potential for making a more effective antivenom if you have more than one format.
And I think that could be an argument in favour of another format other than a vanilla IgG format. I'm remembering Anna Wu talking about mini bodies. I don't know if you remember that, where it was, I think it was a CH3 domain attached to a Fab, so it was smaller than a regular IgG, and I think the argument was better tissue penetration into tumours and so on, but nonetheless with the benefit of a long half-life. So that could be an excuse for a reason for having something other than a vanilla IgG.
Well, just to say for things like tissues and blisters, one of the things we are working on is to consider aspiration rather than even before waiting for these agents. We have a hypothesis that perhaps venom antigen gains entry into the blister fluid, and if you do not do anything, there is gradual slow release of the venom antigen, and we aren't sure that the IgG or the other immunoglobulins gain entry into the blister fluid. Then a year ago, I think a Brazilian group showed in about 2 or 3 patients, yes, the immunoglobulin does go into the blister fluid, but we do not know whether it's functional or not functional. And so clearly for us, clinically, it's better to aspirate it at this stage while awaiting arrival of this newer generation agents that we are sure can penetrate into the blister fluid and mitigate against this depot slow release.
Yeah, I'd like to sort of challenge that your comments about Anna Wu's data, John, because there's this received wisdom that smaller things will penetrate better into tumours, but lots of that's from imaging studies, and what you've actually got is a better tumour to blood ratio at early time points because you get more clearance from just because things are below the renal threshold and clear quickly. But after a few hours, antibodies are much better of penetrating unless they're high affinity with a binding site barrier, because they're protected from degradation. And because by having FC on them and binding to FcRn, they can get better out of the circulation. They are designed to sort of go everywhere. Maybe having a monovalent one could give the advantage that you wouldn't, get sort of high affinity binding at barriers. But something like an antibody, I still think, is the best format in the long run. And if you look at some of the early work that Dame Wittrup did with modelling penetration, you had to be well below the mini body type range. And even with DARPins, which are very small, they're much smaller, and I guess VHH is the same. They get more of an opportunity to go through, but in the end, you still get more in with antibodies.
The thing I would say back in return to that is that these are two very different situations. One is a tumour that's going to be around for quite a while, and therefore one can perhaps afford to take the time to let the IgG penetrate, versus a situation where there's tissue necrosis happening, or so therefore trying to get something in there in the shorter term may be more beneficial than waiting around.
Tissue necrosis is such a big problem anyway because the vasculature is going to be broken down there. So I think these things are really, really complicated to look at, knowing where the disease actually is, and maybe even do some experiments or predictions with modelers to say, what's going to go into those necrotic areas? What can actually get there? And can enough get there to actually do the neutralization? They're really, really big questions, as you know. Sorry, I just get really heads up about this.
No.
Actually, we've done imaging studies, and this is tumours again, but you can really see things going, radiolabel things right into those necrotic centres. Seem to be really good at getting in there, yeah.
So also just I think it's a super good question, and I think just one thing also that's relevant, especially for us that are in the field, to think about is that currently when we look at FABs or IgGs or FAB2 and whatever, we look at typically in the field something from a horse, where if it's the full IgG, where the FC region doesn't probably interact so well with the human receptors. So I think that if we use whatever molecule, but with the human FC, potentially, and I'm not an expert on this, but you could maybe get IgG or IgG-like molecules that would more easily penetrate in, because they're more compatible. That's my guess, but there's probably someone, I know there's someone in the, she was here earlier at least.
And it might, there might be differences, because maybe if you need to have an immediate activity, I think as shown in your model, within minutes, then maybe you need something very small. And in some other instances, you might something that's more long-lasting. I think we had a question over here.
You mentioned or hinted at some of the regulatory challenges maybe in the future, and you've discussed a little bit about the challenges of placebo-controlled trials. Given that MABs for anthrax were approved with the animal rule by the FDA, is a solution better animal models?
I don't think so, but...
Well, I think that's the first thing we desperately need, actually, is better animal models, because our animal models don't reflect snakebite envenoming. So there are huge gaps there. So I guess there's big questions, and this is just for antibodies, but for any new therapeutic, and actually existing therapeutics, we use an animal model that really doesn't reflect the PK of the venom, let alone what the treatment. And we talked a bit about the co-incubation and challenge models, and I think challenge models is definitely moving things forward. But from my perspective, I'm pretty keen for the community to develop completely new models that will allow us to be predictive of human, okay, not of necessarily of human envenoming, but will better mimic the situation, so we can use those models to be more informative. How much of an antibody are we going to need for snakebite, of a monoclonal? I don't think we have a model that can predict that currently. Our animal model can't do that.
Yeah, I disagree a bit with Nick here. I think if you think about completely different modalities, yes, then maybe the animal models are not great. But if you're thinking about proven, known formats such as human IgGs, does it matter whether the mouse spins around of itself or whether whatever the endpoint is? Does it really matter as long as you can make it go away? I mean, if you know that the antibody binds and you've shown it doesn't cross-react to something weird, I don't think that for antibodies, if we talk about human IgGs or proven scaffolds, that we need better animal models. I'm not saying it would be bad to have. I'm just saying efforts, in my opinion, could be spent better elsewhere. But for completely novel modalities where an animal might metabolize it or something, maybe there's an issue, yes. But proven antibodies, I don't think so.
I guess I'm coming at it from a slightly different perspective rather than about the antibodies. It's more a case of, does the model reflect envenoming? And the assumption there is that what you're seeing in the mice is what you would see in a person. We know that's not true. So there's lots of examples where we're measuring an effect of a venom that is not the same effect you would see in a person, and we're measuring neutralization of that effect that is not. So yeah, I think a bit of work probably all round, to be perfectly honest. But I think we definitely can. We will need to rely on animal models because of all of the challenges we have with clinical trials. So we should really be improving those at the same time as all of the other work. And this is a community effort, obviously. I think it would benefit everyone.
Yeah, I think we are sort of nearing the end of the discussion, but I think you all made a very compelling argument. Very compelling argument that we all need to start working on snake antivenoms. So maybe I'll just give one more chance to the panel to sort of give us some final wise words and to get on our way.
Yes, now I got stuck on this animal model thing, so I don't know if there's any wise words to say. I will agree that maybe looking beyond just antibodies and neutralizing toxins, where I do think, yes, we kind of know what we need to do. At least I feel that I know enough to go forward. It would be good to have better models to sort of mimic what then happens generally after you might have neutralized, because there's many of these effects from venoms that even if you do neutralize them halfway, how does the skin or the tissue regenerate? How do we generally treat a snake bite when we don't just think of the antivenom, but auxiliary treatments? And there I would completely agree with Nick. There we can't look at the animals. There we need better models. So maybe, hopefully that's a little bit more balanced, that there's a lot of challenges here all around, and it's a community effort to figure out venom variability, what toxins to go for, how to neutralize them, how to then deploy things, how to train people, how to put things through regulatory affairs. It's complicated.
Yeah, I just think it's great to have this session here today at this conference, and for so many of you to be here and sit and kind of listen to us in the snakebite field talk about the challenges we face, and hopefully some of you get the cogs whirring from your perspective as to the challenge we have and the complexities as well. I mean, there's a lot of complexity out there about snakebite, and there's a lot of challenges that we face, but I think well, you said it, John, you could take a step on the path, that's the only way to begin doing this. And I think in recent years, there's been a lot more attention around this issue than we've ever had in the past. So we kind of have a half glass full hat on at the moment.
That's quite a difficult question. Give us some wise words. If I knew you were going to ask that, I would have saved some up. But I guess I'm a new kid in this field, and I know that there are people who've been working for decades who are not here, who we should give due credit to the efforts over the years. But what's transparently obvious to me is that the bit that I think that we're good at is all doable, and this is just a multi-dimensional, multi-decade problem that's going to involve the efforts and the cooperation of people sitting in regulatory authorities and then all the sort of downstream work. So there's lots of challenges, of which this is arguably one of the easier ones, the discovery.
Okay. First, I would like to say thank you for the invitation. Also, I would like to do a comment regarding the mice models, because as immunologists, we also have problems with mice models, not just for envenomings, for arthritis and multiple sclerosis. So we have all experimental models. So we have to consider the same, I think, for envenomings. But before finish, I would like to ask you to look for snakebite envenomings as an important disease. I'm asking for you here in the audience, like most of you are from companies and universities that work with the discovery of antibodies, and it is an important disease. So you are working with autoimmunity, with cancer, but as I show, the incidence of snake bites in a specific region, that is in Roraima, it's higher than incidence of new cases of cancer. So that's why I'm pretty happy to be here to talk about our issues in our country. And I hope that now from this panel, you also look with another look to this important neglected problem.
Thank you. Basically two points. One, to reiterate the fact that snakebite is a cause of death, disability, disfigurement, and deprivation, those 4 Ds. And second, solution for it, access. I think the most important thing we should be looking at, how do we improve and facilitate access to the widest number of victims of snakebite? One, we need to reduce and curtail delay. Delay is a major issue. If we can improve funding and financing, I see a situation where we'll improve access to the ones that really need it. So as we go out there, let's explore how can we improve access. And related to that, we are battling with polyclonal antibodies now, that they are being very expensive, prohibitive, not affordable. How do we envisage new generation recombinant monoclonal antibodies? How can we force the price to be affordable to those that need it? I believe many companies, you could serve as ambassadors to somehow get into corporate social responsibility, or get a platform at the global level wherein access can be promoted, funding for snakebite can be explored, so that the widest number of victims can benefit from this. Thank you.
Okay, thank you very much. So give this panel a great hand, and then